We're going to go for the next session in this program, which is the presentation by the winner of the Young Investigator Award for 2013, which is Dr. Maria Llorens Martin, who's going to talk us about her paper titled GSK3 Beta Overexpression Causes Reversible Alteration on Postsynaptic Densities and Dendritic Morphology of Hippocampal Granule Neurons in vivo. So Dr. Avila and Maria Angeles Perez will present the award at the end of the presentation. So Maria, please.
Okay, first of all, I would like to thank the jury for the Cybernet Young Investigator Award for having bestowed on me not only this award, but also the opportunity to share our work with all of you, a work which has been done in Jesus Avila's lab, and of course, many thanks to all of you for your attention. From a general point of view, one of the main goals of our group is to understand the molecular and cellular mechanisms leading our brain from a physiological state to Alzheimer's disease, in order to find out to what extent these alterations could be reversible. Among all the molecules whose activity is dysregulated in Alzheimer's disease, one of the most interesting to us is GSK3 beta, since it is considered the molecular link between amyloid beta and tau. In order to further investigate the involvement of GSK3 in the pathogenesis of the disease, a conditional murine model overexpressing GSK3 under the control of the neuronal promoter CAMK2 was generated in our lab several years ago. As you can see in this representative image, in this mouse line, GSK3 overexpression takes place mainly in the hippocampus. The administration of doxycycline permits a selective control of GSK3 overexpression, which can be visualized by immunohistochemistry against the reporter beta-galactosidase, as you can see in this image in white color. We had previously demonstrated that this animal model exhibited most of the typical features of an AD model, such as an impaired LTP generation, altered spatial learning, hippocampal atrophy, and importantly, a strong brain pro-inflammatory phenotype. These features altogether outline a general lack of neural plasticity, which may have functional consequences at different levels. But what does the term plasticity mean in the context of the central nervous system? In general terms, plasticity can be defined as the capability of being shaped or formed. Thus, one can say that neural plasticity is the ability of our nervous system of being restructured, thus becoming adapted to the environment and to the experience. However, there are multiple levels or types of neural plasticity, ranging from structural plasticity, which takes place, for instance, after a lesion, to synaptic plasticity, which takes place at the level of the individual synapse. However, one of the most drastic types of neural plasticity is the birth of adult neurons in the adult brain. This is adult neurogenesis, and this process involves the integration of new synaptic elements into a pre-developed circuit, thus sharing features with both structural and synaptic plasticity. Among the different methodological tools to study adult neurogenesis, one of the most useful tools that has arisen in the last decades is retroviral labeling due to their ability to label only dividing cells. As you can see in these representative confocal images, GFP-expressing retroviruses allow the visualization and the analysis of newborn neuron morphology, whereas PSD95 GFP-expressing retroviruses allow the analysis of the postsynaptic densities in terms of their volume and their number. Since adult hippocampal neurogenesis alterations are present in most of the AD animal models and also in AD patients and may help to understand the phenotype of our mice, we have used various retroviral vectors in order to study the alterations that GSK3 overexpression could produce on newborn neurons. But firstly, we wanted to characterize the neuronal subpopulations overexpressing GSK3 in our model. To do that, we injected GFP-expressing retroviruses into the hippocampus of GSK3 overexpressing mice and sacrificed the animals after different survival times. As you can see in this graph and also in the images, we could determine that GSK3 overexpression took place for the first time in 3 or 4 week-old neurons. Accordingly, nor glial nor neuroprogenitor cells were positive for beta-galactosidase. But in contrast, most of the double-cortin positive neuroblasts and most also of the new and positive mature granule neurons were positive for beta-galactosidase. This way, we could distinguish between those cells being directly affected by GSK3 overexpression and those cells in which indirect, non-cell autonomous were taking place. Next, in order to study the alterations that GSK3 overexpression could produce on newborn neuron connectivity, we injected PSD95 GFP-expressing retroviruses into the hippocampus of GSK3 overexpressing and wild-type mice, and again sacrificed the animals after different survival times. Most interestingly, what we found was that GSK3 overexpression produced drastic alterations in newborn neuron connectivity. In these graphs, you can observe how GSK3 overexpression reduced not only the number, but also the volume of the postsynaptic density. In this second graph, we have represented both the volume of the postsynaptic cluster and the percentage of clusters. Thus, you can observe how GSK3 overexpression produced an increase in the percentage of small postsynaptic clusters, thus supporting the predominant role played by this kinase at the synapse. In addition, GSK3 overexpression drastically altered newborn neuron morphology. In these images, you can see how a typical granule neuron obtained from a wild-type mouse display one single apical dendrite emerging from the soma, what is extensively branched in the molecular layer. In contrast, GSK3 overexpressing neurons display more than one primary apical dendrite. And here in the quantifications, I wanted to show you how in this Show's analysis graph in which we have represented both the number of branches and the distance to the soma, GSK3 overexpression produced an increased proximal branching of the dendritic tree, whereas distal branching is significantly reduced. In this other graph, we have represented both the total dendritic length and the cell age. And you can observe how one of the most striking results was the fact that we found morphological alterations in cells younger than 4 weeks of age, in which GSK3 overexpression was not yet taking place. This points to the existence of some indirect effects which are related to the pro-inflammatory phenotype observed in these mice, as we have already demonstrated in a subsequent work. Nevertheless, what is most relevant is that after performing a comparative morphometrical analysis between mice overexpressing GSK3 and human patients of AD, we found remarkable morphological similarities between both types of granule neurons, and that morphology is drastically different from that of controlled patients. In the graphs, you can observe how granule neurons obtained from AD patient display more than one primary apical dendrite and also an increased proximal branching, whereas distal branching is again reduced. These morphological features, as you will remember, are quite similar to those observed in GSK3 overexpressing mice. Subsequently, we tried to reverse these cellular alterations produced by GSK3. To that aim, we injected the retroviral vectors, and 4 weeks later, we divided the animals into 3 experimental conditions: a control housing group, a doxycycline group in order to switch off GSK3 overexpression, and finally, an environmental enrichment group as a group to increase hippocampal plasticity. So in this video, please could you play the video? Okay, thank you. In this video, I want to show you how environmental enrichment ensures a higher physical activity, an increased social interaction, and also a maintained cognitive stimulation due to the presence of multiple objects and toys which are being constantly changed. So the question was, are the cellular alterations produced by GSK3 overexpression reversible? And as a tentative answer to that question, we may say that both environmental enrichment and doxycycline treatment were able to completely normalize the aberrant morphology produced by GSK3 overexpression. In the graphs, you can observe how both environmental enrichment in green color and also doxycycline treatment in purple completely normalize the aberrant branching of the dendritic tree in both the proximal and the distal domains of this dendritic tree. But what is most relevant, can also the alterations in connectivity be reversed? Here you can observe how both doxycycline treatment in purple and environmental enrichment in blue are able to normalize the reduced number of the postsynaptic clusters produced by GSK3 overexpression, completely in the case of doxycycline and partially in the case of environmental enrichment. However, the analysis of the volume of the postsynaptic cluster revealed that both strategies were acting through different mechanisms. First, in green color, you can see how environmental enrichment significantly increased the percentage of small, newly generated synaptic contacts in wild-type mice, but significantly increased the average volume of the postsynaptic density in GSK3 overexpressing mice. However, after switching off the transgenic system, the addition of small postsynaptic clusters is possible again. This way, it is possible to conclude that both environmental enrichment and GSK3 overexpression switching off can reinforce dentate-gyrus connectivity by acting through different mechanisms, and hence that the alterations in connectivity seem to be somehow reversible in this animal model of the disease. And finally, just to sum up, we have proposed the following schematic model. GSK3 overexpression provokes mature granule neuron death, a phenomenon that activates microglia and the release of pro-inflammatory cytokines. This phenomenon compromises newborn neuron maturation, whereas GSK3 overexpression reduces the number and volume of postsynaptic clusters. Doxycycline treatment abolishes GSK3 overexpression and consequently brain inflammation. This way, newborn neuron maturation is normalized and the addition of new postsynaptic contacts is again possible. Environmental enrichment increases the level of neuroprotective and pro-survival factors and normalizes the levels of pro-inflammatory cytokines, promoting the addition of new postsynaptic contacts in wild-type mice, but significantly increasing the volume of the pre-existing synaptic densities in GSK3 overexpressing mice, thus reinforcing dentate-gyrus connectivity through this alternative mechanism. And just to finish, even though all of you may know them already, I would like to introduce you the group in which I work as a postdoc. It is Jesus Avila's lab. And I would like to thank all those persons who have made this work possible, especially Jesus for his continuous support and enthusiasm, and for having allowed me to develop this project in his lab, and also Almudena Fuster Matanzo, Alberto Rábano for having provided us with the human samples, and especially Katia Teixeira for having performed all the retroviral injections of this work. And finally, many thanks to all of you for your attention.
All right, so the paper is open for a couple of questions, if there are any. No questions? Yeah.
Thank you. That was a very nice talk. And of course, I'm interested in, on the role of neuroinflammation in your model. Did you have any experiments, or did you try to track down the responsible cytokines, or did you test what could be, maybe it's complement or a chemokine? Any idea?
Yes, that's a very interesting question. We have already published another paper. Well, it's accepted for publication. It will be online maybe this week or next one in Translational Psychiatry. And we have demonstrated on one side that administering LPS to the mice produced these same morphological alterations in the granular neurons. And on the other side that ibuprofen treatment to these mice completely normalized the aberrant morphology. However, this is the positive part, but we have the negative part also, and it's that administering ibuprofen to these mice reduces even more the number of postsynaptic clusters. So what we have found is the microglia in these mice is not as sensitive to ibuprofen to block neuroinflammation as it is in a pure model of neuroinflammation as LPS model. So it seems that this microglia that in fact expresses a different pattern of cytokines, we have made an array and we found an M2 phenotype just overexpressing many types of cytokines, both neuroprotective and also inflammatory. And this microglia for sure should play a different role, an important role in controlling both synaptic plasticity and also the morphology.
Thank you, very interesting.
Thank you.
All right, so if there are no more questions, we're going to proceed to the presentation of the award by Dr. Avila and María Ángeles Pérez Muñoz.
Bueno, ahora vamos a empezar el claustro y el claustro va a empezar la gerente de Cibernet, María Ángeles, y después voy a continuar yo un poco, ya cerraremos hoy ya la sesión que ha sido muy larga.